All right, everybody, let's talk about our results from the Cultivation of Anaerobes lab. So if you recall, what we did is we took some control samples. We also took our unknowns and we inoculated some BHI plates and we inoculated some thioglycolate broth tubes. And the purpose of this lab is to really examine the requirements for oxygen in these microbes' growth. Okay, so anaerobes, so aerobe meaning, oxygen, and an meaning not. So in other words, this lab is kind of alluding to things that don't need oxygen. So we assume, we talk about animals and life and stuff like that, that we certainly need oxygen. Other animals need oxygen, even fish, things like that. They get the oxygen from the water through the gills, but microorganisms, maybe they need oxygen, maybe they don't, and it depends on what's going on. It depends on their particular niche where they occupy and things like that. So not all is like us, as one of these things this is alluding to. And again, this is a part of these labs where we are examining the growth conditions, the different things that allow or prohibit or letting these microorganisms grow. Okay, so cultivation of anaerobes. So we took our organisms and we grew them in these different situations. Then for the plates, we put them in these brewer's jars, and that was an oxygen-free environment. We let those grow, and we then did the thioglycolate broth. So this thioglycolate broth is a really interesting kind of thing because it's a liquid media. It is differential, remember, and so differential is it's going to tell us information. It's not going to prevent things from growing necessarily, but it by itself is differential. And so we had that tube, and it had a nice screw top. And so the uninoculated tube, well, it had this kind of like bluish, greenish top. And then the rest of it was this kind of yellowish tan color. Right, so this was our uninoculated thioglycolate broth tube. And so we were thinking about this, well, what does this do? What is the point of this tube? Well, up here at the top, and down here at the bottom, and even here in the middle, they kind of have different oxygen availability, right? So if we're talking about oxygen or O2, like the oxygen gas, think about where in this tube is the most oxygen, where is the least. And that is the whole purpose of using the tube. So just think about that for a second. Maybe we've gone over this before, maybe not. But think about it. Remember, these are the thioglycolate broth tubes, and there's different amounts of this oxygen available. And so that's going to dictate maybe what happens in the tube. Okay, so if you can imagine that there's different amounts of oxygen in this tube, then that would kind of let you know maybe what will happen is organisms will grow in different places. So maybe as you imagined that there is the most amount of, I'm going to get rid of the pencil, maybe there's the most amount of oxygen up towards the top. And then down at the bottom, there's like no O2, right? So basically, within this tube, we have an oxygen gradient of high to low. And then where our organism can grow is going to let us know about that individual organism's oxygen needs. Okay, so the question is, like, where does my organism grow? And then what does that tell us about the oxygen needs? So, well, let's say we inoculate our tube. And remember, the blue up here, that's methylene blue. It's an oxygen indicator. So as long as that's blue, that means there is O2 present. And that will go away if the oxygen gets used up. And you could imagine that the oxygen would get used up by an organism, well, that uses oxygen. So let's say we inoculate our tube, we let it grow for a period of time, and we come back, and all the growth is down here at the bottom. Right? There's just like a little pellet of growth down there at the bottom. What is that telling you about the requirements or the needs for that organism? Or conversely, again, what if we have growth just up here at the top, like a little floating ring? What would that tell you about its growth? And if that organism is growing up there, you're probably not going to see that blue up there. Okay? So just kind of think about that, make your predictions of what you would expect to see. And now let's talk about some different categories of growth that are important to us. So there's basically 5 categories that we need to think about. And these are kind of illustrated in your book, but not the best way. So we'll just go through here. Pardon my drawing. So here are 5 different thioglycolate broth tubes. And in our book, or the 5 we really want to think about, are some that are facultative, and facultative means can have, can use, and then some that are obligate. And if something's obligated or obligate, that means it must have, okay? So that's a little bit of notes in there to keep. Facultative means can use, kind of, and obligate means has to have. Well, so let's start with some easy ones. Let's start with an obligate. Let's start with 2 obligates, actually. And so there's 2 opposites here, right? We would have our obligate aerobe, meaning must have oxygen, or an obligate anaerobe. I think I'm going to change my, there we go. Obligate aerobe and obligate anaerobe. Okay? We would probably expect the growth to be completely opposite in that slide before. So the aerobe, where do you think those grow? In other words, aerobe must have oxygen to live. Well, I would imagine in that tube it would be growing at the top. Maybe actually what we should do, let's fill these in with our media. Magic of colors. All right. So here's our media for each one of these. I wasn't very good in grade school. I couldn't color within the lines. I apologize. All right. So there's our 5 thioglycol broth tubes. Oh, let's not forget our methylene blue layer. Okay. So our obligate aerobe and our obligate anaerobe. So we said the obligate aerobe is going to be up there, and the obligate anaerobe is going to be down here, probably towards the bottom, where there's no oxygen. But let's think about that methylene blue layer really quickly. If this one is using the oxygen, is there going to be much oxygen left? And actually, no. We would not expect the methylene blue to maintain in that tube. So an obligate aerobe would be right at the top, and obligate anaerobe would be all the way at the bottom, and there would still be methylene blue as that oxygen indicator. Great. Let's move on a little bit now. Let's talk about a little aerophile. Okay, philic or phile means loving. Micro means little amount. So a micro aerophile can't have too much oxygen. So up here is too much oxygen, down here is way too little. There's just a very narrow band in which they can grow, and you would see them like just a solid ring. Because remember, there's this gradient going down, and depending on just what they want, you'd see them growing right in there. And they're using just a tiny bit of oxygen, but they are, so they would probably consume. You may see a little bit of methylene blue indicator, but could be all gone because they still are using up oxygen, but just little sips, a little bit, not too much, not too much. Okay, so the next 2. Okay, well, so if they aren't fully oxygenated or no oxygen, and then just a little bit of oxygen, that means these are somewhere in between, but not as specific as this. That's supposed to be an arrow. Not as specific as this. Something in between. Well, so we have that facultative thing I mentioned. Let's put this here. And then we have, so there's a facultative anaerobe. And then this is something that's called aerotolerant. I put a hyphen there. It's all one word, aerotolerant. So that means these are somewhere in between. But what is the difference between these 2? Well, for both of these, you can see growth throughout the entire tube. Okay, in other words, an anaerobe in the name, that means you could have growth down here at the bottom. You can have growth up here at the top. Aerotolerant, you can have growth down here at the bottom. You can have growth up here at the top. Well, so what is the difference? Well, aerotolerant, we'll start with that one. Aerotolerant is just what the name says. It tolerates air. But it doesn't use it. It kind of could care less. So you would see growth throughout the entire tube. Just an even spread. depending on how it was inoculated and things like that. But it could be just a whole cloudy tube. But basically, from the top to the bottom, it is possible for that organism that is aerotolerant to grow. Facultative anaerobe, however, it does not have to have oxygen, so it is capable of growing down here, and is capable of growing throughout the entire tube. But when there is oxygen present, it grows much better. And so you would get a lot of growth at the top, and then kind of less throughout. And sometimes in these you see this little like tornado shape from the inoculation site. But you typically would have a lot of growth at the top. So if you held this up to the light, you'd see a very thick disc at the top. And then throughout, just regular growth. And since the facultative anaerobes can use the oxygen, you would not see methylene blue. Now, so what is going on here? Well, these facultative anaerobes don't have to have the oxygen, hence their ability to grow down here. But when oxygen is present, they are able to use other metabolic pathways other than fermentation, which gives them more energy and the ability to reproduce better. It gives them, they can use it as a resource. Therefore, the ones up here have more resources than the ones down here, so that's why you have a much higher amount of growth. Aerotolerant, on the other hand, the ones up here are just doing as well as the ones down here. Because oxygen, whatever, we can tolerate it, it doesn't kill us. But these obligate anaerobes are killed, actively killed by the oxygen. Because remember, oxygen is this, it's a molecular, it's a molecule, it's a gas, and it can actually tear apart molecules. You hear about oxidation, oxidation reduction accident, oxidizing agents. Oxygen can really pull apart molecules. And if you don't have the proper enzymes or metabolic pathways to handle too much oxygen, well, like an obligate anaerobe, it would kill you. And then the other side of that, these obligate aerobes, well, they only can produce energy in the presence of oxygen. And so we see these, this distribution. So these are the 5 we should definitely be familiar with. Obligate anaerobes, obligate aerobes, microaerophiles, facultative anaerobes, and aerotolerant organisms. So that was our thioglycolate tube. We also, though, did plates. I don't know if there might be a better way to do this, but we're just going to erase. We also did plates. And the plates we grew in the presence of, plates with no oxygen present. Well, where are the ones that we grew with oxygen? Well, if you remember, we did our unknowns, and those have been in the refrigerator for a while because we did colonies with those a long time ago. We did streak plates with them. So you have your 2 plates. And if this was with oxygen, right, like the ones we did our streak plates on, and this one is without, well, what would you expect? If your organism is obligate aerobe, well, you'd see growth right here. If your organism was an obligate anaerobe, you'd only see growth here. Microaerophile would grow here, but maybe not as well. But now, these facultative anaerobes and these aerotolerants, they would grow on both plates. You could actually possibly, depending on how your plating is, you could maybe tell the difference between a facultative anaerobe and an aerotolerant if it grows better in the open air plate versus without. But, we didn't really control for the amount we were plating and things like that, so this might be difficult to assess. So there you go. The plates were just kind of an on and off binary thing, and pretty much all of our unknowns, because we knew they grew in the oxygen environment, and most of them, pretty much all of them, all of them, are going to grow without too. So your unknown is going to be a facultative anaerobe or an aerotolerant, and you just have to make kind of make that decision based on how it grew in the tube and things like that, and figure it out. Okay, so those are our cultivation of anaerobes lab. So make sure you're familiar with those 5 categories we talked about. You might not have data for your unknown, and so if that's the case, you have to kind of do some reverse engineering once you figure out what your own unknown is, and then you can kind of do some research and figure out the oxygen requirements. But that should be good, and so that's the cultivation of anaerobes lab. Thank you very much.